Hey honors, today we're going to be learning about complex inheritance patterns. So you took notes on Monday over all of these topics. I'm just going to be explaining pretty much all the slides of the PowerPoint and writing out some notes. So today we're doing complex inheritance patterns. All right. Let's see if I can focus that any better. That made it worse. Let's try it again. No. There it goes. Behaved. Okay. Complex inheritance patterns. So the notes started off talking about Mendel's laws. We had the law of dominance. That just means there are some traits that overshadow other traits, and we call those overshadowed traits recessive. So if there's ever a presence of a dominant letter, then that's going to overshadow the recessive trait. There's also, let's see, the law of segregation. And the last one was independent assortment. All right, so for the law of segregation, so both of those, these last two laws are demonstrated in Punnett squares. So what the law of segregation says is a person might have, a person has a diploid genotype, but when they go to form their sex cells, their sperm or their eggs, those allele pairs segregate, they split up. Right, so they don't stay together when they become sperm. So in the person, they're together, but when they're made into sperm, they are segregated, they're separated. And then independent assortment is demonstrated by how those get together on the inside. So independent assortment is being demonstrated inside the box, and segregation is on the outside of the box. So how do they assort? How do they get back together? And what that means is that these are going to be inherited independently. This sperm acts independent of this sperm. Even though at one time these genotypes were together, once they're segregated into different sex cells, they're now independent of each other. So that was those laws. But we're going to kind of leave Mendel behind, and we're going to talk about some more complex scenarios. So the first that the notes talked about was this concept of incomplete dominance. So this is when you don't have Mendelian dominance. This is when traits are blended. Let's see. Let's go down here. Example. Gosh, I should have thought these out. Would be red mixed with white will give you pink offspring. Okay, so let's say you have a red flower and you cross it with a white flower. Okay, you are going to get all pink flowers. So if we do the Punnett square, You're still going to do the Punnett square in the same way. But in this case, we have 100% pink offspring. So this would be an example of incomplete dominance. A week ago when we were doing just Mendelian or complete dominance, these offspring would have given you the red phenotype. But this is a special case. This is a case where sometimes genetics don't follow the Mendelian laws, and sometimes you'll get this blended phenotype instead. That's an example of incomplete dominance. Another exemption to the Mendelian rules is called codominance. This is when you get both traits expressed. This could also have multiple alleles, so more than just one letter. And the example for this is blood type. So for blood type, you could be type A, type AB, type B, or type O. And when we talk about our blood type, what these letters are actually referring to. Okay, so this is a blood cell. And on the surface of all of our cells, but on these in particular, we have these little glycoproteins. They're like little receptors, glycoproteins. So they're little sugar chains attached to a protein chain. And they can have different shapes. And depending on what shape you have, determines what type of blood type you have. So this would be the B receptor, what it looks like, and this is what an A receptor looks like. So this person would have AB blood, right? If we looked at another cell, this is from someone else, you'll notice they only have A receptors. So this would be A type blood. All right, we're going to do another one. This would be B type blood. And then the last cell is O type blood. And you'll notice that O has no receptors. So there are no receptors on O. So to have the different blood types, you could be AA or AO, and that would be A type. You could be BB or BO, and that would be B type. AB is AB type. And then to be O, you have to be double O. All right, so those are the different genotype combinations you can be to have the different blood types. So let's do an example cross. So let's cross AB with BO. All right, so if we do that, mom has AB blood, dad has B blood. Their offspring, they have a 25% chance of having AB, 25% chance of having A, and a 50% chance of having B type. So we got 25% AB blood, 25% A blood, and 50% B. All right, so that's how you do that cross. Sometimes they're written as I's. So you would do this in the same way. So so let me show you that over here. So it might look like this. Either way you want to do it, it's the same thing. Either use the I's, like the individual has this blood type. I really don't understand the history of why these crosses look like this. But this, I'm going to do the same thing over here. So we're crossing AB with a BO. So okay. And so we're going to get the same results that we got over here because it's the exact same cross. Okay. All right, that is blood type. What else we got? So that's multiple allele codominance. We did incomplete dominance. Let's go through polygenic and linked, and then we'll do sex-linked. Polygenic. Polygenic is the God's honest truth for most of our traits. So polygenic just means many genes involved. This is for traits that have a spectrum of phenotypes. So height, for example, right? Because we have right, you could be 4'10", 4'11", 5 foot, 5'1", 5'2". Ooh, I did that one wrong. You could be 5'7", 5'9", 6'4", etc. So we have a bunch. This is a whole spectrum of heights, right? And it gives us this like bell curve. And we'll say 5 foot down here, and we'll do 6 foot up here. So we have all of these different heights. We don't just have 3 heights, right? It's not like there's only 3 adult human heights you can be. You can be 5 foot, 5'5", or 6 foot, and that's it. So that's why this is polygenic. There's a bunch. It's a whole spectrum. Eye color is also polygenic inheritance. So that means we're going to have, so it's not really dad gives you a big B, mom gives you a little b. They actually give you like 4 or 5 different alleles. So there's probably 10 alleles involved in eye color. To do Punnett squares, to keep them simple, we always say like, oh, dad gave you a big B, mom gave you a little b. But that's not the truth, right? Because eye color can be everything from gray to blue, green to green, hazel, brown, and on. So it's a whole spectrum of colors. It's not just you have, and then even within blue, right, there's a range. You can have really like, pale blue, you can have dark blue, same with green, so there's a whole spectrum, within these. It's not just 3 eye colors, your blue eyes or brown eyes. It's not really true. There's a whole bunch of eye colors. All right, that's polygenic inheritance. Another vocab word they went through was linked genes. This refers to like genes that have a tendency to be inherited together. So traits that are typically inherited together. Example would be, red hair and freckles. Or for for dogs, it might be black fur and black nose. Like typically those two traits are inherited together. If they have black fur, they probably also have a black nose. If they have like yellow fur, then they might have more of a pink nose. So those would be inherited together. So those are linked genes. And what that refers to, if we go back to our chromosome, they are traits that are, right, this might be freckles, and this is the gene for freckles gene, and this would be the red hair gene, okay? They are physically, so the genes are physically close together on the chromosome. And so, if crossing over was happening, they would be inherited together. If they were far apart, genes are not linked. Not inherited together. Okay, so that's linked genes. Okay, the last thing we're going to talk about are sex-linked traits. Kim, you want to say hi? My cat just jumped up here. You want to say hi? Come over here. Show me your pretty girl face. You want to say hi? I can force her under the document camera. Kim. Okay, there you go. She said hi. All right. Sex-linked traits, also referred to as X-linked, because they're traits that are carried on the X chromosome typically. So, but these are referring to traits carried on the genes of the sex chromosomes, which you remember is the 23rd pair. XX gives you female, XY gives you male. Let's take a look at those two chromosomes and why this type of inheritance is referred to as X-linked. Hey, bud. Okay, well, I have to finish this video.
How much longer?
15 minutes.
Okay. Yeah, it's fine.
All right, so this is the X chromosome, and you'll see that it's much, much bigger. This is the Y chromosome, and you see how it's pretty small. Purple's not showing up great. So the X chromosome is bigger, and it carries 1,500 genes. The Y chromosome is really small, and it carries maybe 72 genes, so like under 100 genes. Okay, so why that matters. So 1,500 genes can give you a minimum of 1,500 proteins, whereas this might only give you like 70 proteins. Why do we care about proteins? Remember, proteins give us our traits. That's what gives us our phenotype. Proteins give us our phenotype. What you're looking at is proteins expressed in different people. Okay, so the X chromosome carries a lot more traits than the Y chromosome, and so that's why this is usually called an X-linked trait. So let's give you an example. So we're going to use hemophilia as an example. Hemophilia is a blood clotting disease. Hemophiliacs' bloods do not clot, so they have to get a lot of blood transfusions. So you could be, if you're a female, right? So this is a sex-linked recessive trait. So this female does not, this is called a carrier. She does not have the disease. She's just a carrier, but she has one good working X. So she's making the right clotting proteins, so she's fine. Her blood clots. If she was like this and she has no working genes, she's not making the right protein, she has the disease. Okay, guys, because they only get one X, they have a much greater chance of developing the disorder. So this guy here, his X chromosome was good. He inherited a good X chromosome. And so he does not have the disease. But this guy has the disease because his X chromosome, he inherited a faulty one. All right, so let's do a problem example. Let's say this female was crossed with a hemophiliac man. So what are the odds that their children are going to have the disease? So she's a carrier, she doesn't have it. Carrier, and he has it. All right, so what are the odds that their kids are going to have it? So we just do a regular Punnett square. All right. So let's see. So what are the odds that they'd have an offspring with the disorder? They have a 50% chance of having an offspring with the disease. So of the girls, we have one of two girls will have the disease, or 50% of the girls will have the disease, and 1 of 2 boys will have the disease. Overall, their offspring have a 50% chance of having that disease. Alrighty, that is it for those notes. I believe that is everything we went through. I will post the key to the practice sheet that's due today. So, Scouts honor that you actually do it on your own and just look at the key to check your work. That's the best way to do it. But in case some of you are lost or you don't know how to start a problem, I'll just have the key open and available for you today. All right. See ya.